Hi everyone, this is the last lecture video for the semester. Yay! Great job for making it this far for this anatomy and physiology class. It sure isn't an easy class. So this last lecture is not too long and concepts are again pretty straightforward. Since we already talked a bit about the somatic nervous system back in chapter 13, we are going to mention a few more things from chapter 15 about this division of the nervous system, which is somatic nervous system. But the bulk of this lecture will be about chapter 16, which has to do with the autonomic nervous system, which we introduced just a little bit in chapter 12, but didn't really get through it, talk about it in detail. So now we're going to talk about it in this lecture, which is the main system we will continuously refer back to in the second semester of AMP2. So this is the perfect chapter to end this semester with. All right, let's get started. So we have already mentioned previously in chapter 12 that the peripheral nervous system is divided into two main divisions, which is your somatic nervous system and autonomic nervous system. The somatic nervous system usually involves voluntary control of skeletal muscle contraction, whereas your autonomic nervous system controls the smooth and cardiac muscles along with other glands and adipocytes in regulation of our body's homeostasis. So that is why we'll talk a lot about autonomic nervous system next semester, because we're going to talk about all those other organ systems, that's associated with that. Your cardiovascular system, your respiratory system, your digestive system, urinary system, etc. Okay? All right, so we have already talked about the two main pathways of information traveling toward and out of the central nervous system, and we're always sending sensory information toward the central nervous system, which collects information from our sensory receptors, which then generate action potentials within the sensory neuron, which will then send the electrical impulse toward the central nervous system. So we are aware of something we touch that was hot or cold, painful, etc. So remember that this was the afferent division of the peripheral nervous system in terms of the direction of traveling information, okay? So this kind of illustrates and summarizes all that we talked about in terms of sensory pathway, okay? So depolarization at the receptor, action potential at the sensory neuron, and then you have the propagation of the signal from the sensory neuron toward the central nervous system. Okay, for somatic sensory information, they usually travel via the spinal cord all the way up to the sensory processing centers within your primary somatosensory cortex or your cerebellum that we have talked about in chapter 14. For visceral sensory information, they usually travel to the brainstem and diencephalon on the other hand, okay? And then for information coming out of the central nervous system toward the peripheral nervous system, remember we call these in terms of motor pathway or motor informations and commands toward your effector tissues. So usually this involves the efferent fibers of the peripheral nervous system, which I talked about. Afferent, A for arrival, which means sensory information arriving at the central nervous system. Efferent for exit, E for exit, things or information coming out of the central nervous system in form of motor commands. So the somatic motor pathway will control the peripheral effectors such as your skeletal muscles, which may result in voluntary or involuntary actions. So somatic usually go towards skeletal muscles. Okay, some motor commands may be interfered or modified, however, by our higher order functions of the brain, because whenever we're thinking about something, of course, some things is gonna work as a reflex kind of thing without us thinking about it, but some actions definitely require us to plan, to remember some stuff, and also learn. For example, learning how to play the piano, riding the bike we talked about, or planning ahead of time so you have enough time to study before an exam, and memory, of course, remembering all those information from the exam. All those things will require a little bit more from your memory sense or higher order functions of the nervous system. And we will talk slightly about memory at the very end of the chapter. Okay? All right, so let's talk about the sensory receptors a bit, since we have only mentioned a few from the integumentary system and the special senses chapter. But now we're really going to categorize them. So as we have seen from the integumentary system with Meissner's and Pacinius corpuscles, these were cells that are able to send information toward the sensory neurons that they form synapses with. Or in the olfactory sensory neurons case, their own dendrites can serve as sensory receptors to the odorants that we smell. Okay, so usually these can either be sensory neurons themselves, or there are specialized cells that are, representing and performing the job of a sensory receptor. But regardless of what type of sensory receptors they are, their main goal is to provide information about conditions that is either on the outside or on the inside of your body that may require the attention of the central nervous system to react in response, or at least for us to be aware of that sense coming through. Okay. So arriving information is called sensation, while the conscious awareness of the sensation is called perception. Okay, remember from the withdrawal reflex we talked about in chapter 13, we saw that the interneuron that relayed information between the sensory neuron feeling that hot pan and then the motor neuron of lifting your hand away from the hot pan, that interneuron had a separate collateral that extends up to the brain to give us that sensation of pain. And hence, this is why I talked about, with a lot of feelings that we have from our sensory receptors, they have to reach the brain in order for us to be consciously aware of the sensation. And that part of it is called perception. Okay. So the the conversion from an arriving stimulus to an action potential propagated toward the central nervous system is called transduction. We didn't really give that an official name when I was talking about all these pathways in chapter 17, but that whole pathway from your sensory receptors all the way being able to arrive at your brain at particular specific destinations, that whole pathway is called transduction. Okay, a lot of these kind of sounds like electronic stuff, okay, which is basically what it is. It is these electrical impulses that's being propagated from one cell to the next, okay, whether it be a receptor cell or a neuron or a neuron tissues like the brain. So there are two main categories of sensory information. Our general senses pertains to sensitivities to temperature, pain, touch, pressure, vibration, and even proprioception, while our special senses has to do with what we talked about in chapter 17, which is olfaction, gustation, vision, equilibrium, and hearing. These are usually involving more complex receptors, which was why I chose to talk about that before this chapter, because chapter 17 is a more complex chapter involving a lot of stuff for those special senses. So we talked a lot about those special senses from chapter 17. So now we will expand on the general senses here from what we learned in the integumentary system, because we just mentioned it briefly with those Meissner's and Pacinius corpuscle, but there's actually a lot more different types of receptor that has to do with those general senses. Now, in terms of a sensory receptor, their cells usually have a limited area for which they're serving to receive information from. Now, it's very rare that one single particular sensory receptor cell can monitor the whole surface of the entire body. And that's just not an accurate way of being able to localize your source of sensory information pathway. But the area that the sensory receptors are receiving sensory information from is what we call a receptive field. And, they're kind of like our little GPS navigation circle that help us pinpoint where the sensory information is coming from so our central nervous system can send the response to the correct location, or for us ourselves to be consciously aware of where that certain feeling, if we can be aware of it, where that sensation is coming from on our body. Some we can sense, some we can't, okay? But for example, like someone pinching you or tickling you, you can feel those, and you can tell whether someone's tickling you around the neck versus someone is tickling you around the back or your waist. So that's due to having different receptive field that a certain sensory receptor is serving for, okay? And the smaller the receptor field is, the more accurate we're able to localize the source of the stimulus, or the other way around is true as well. The larger this receptive field, the harder it is to localize the stimulus. And that's why I said if we only have one receptor cell for the entire surface of the body, so you will never be able to tell. If I poke your arm, you would think the whole body is being poked, okay? You won't know that I'm specifically poking at your arm, is because you have to have different receptive field in order for you to know and localize that particular source of stimulus, which is me poking your arm, okay? So, for example, if we think about receptive field of our general body surfaces, they are about as big as 7 centimeters, which is pretty large if you think about how long 7 centimeter is, whereas those on your tongue and fingertips are less than 1 millimeter long. Okay, so if you remember a little metric ruler and you see centimeter versus millimeter, those millimeter little tick marks are those tiny little tick marks on your ruler. So one receptive field for those sensory receptors on your fingertips is smaller than that one little tick mark, okay? So this is why we are able to feel so much better with the tips of our fingers and versus the rest of our body. And sometimes we can even tell what the substance is just by feeling the object without having to see the object. For example, if someone placed an orange versus an apple on your back, okay, while you're laying down, of course, you probably can't tell the difference between the two. It's just some very cold fruits that's on your back, or round thing that's on your back all of a sudden. But if you were given the orange or the apple in your hand and you're able to feel it with your fingertips while blindfolded, you probably can tell which fruit it is that is in your hand. You probably can tell this is an orange or an apple just by feeling those different roughness of textures on the surface of the orange and the shape of the orange versus the shape of the apple. So this is what having a receptive field size means, okay? The smaller it is, the more accurate you're able to tell the details or where that source is coming from. The larger it is, the less likely you're able to tell. The other thing we used to do in anatomy and physiology lab is we will have 2 toothpicks where we poke, and then we kind of measure how big a receptive field is by poking your fingertips, okay, poking your fingertips with the 2 toothpicks versus poking the 2 toothpicks on your back. And you will find that we start from far apart of the 2 toothpick, maybe 10 centimeters apart, and we'll move it closer and closer and closer until a point that the subject says, oh, I can't feel the difference between the 2 toothpick anymore. That's how you can measure your receptive field. And what you will find is that those on your fingertips, you can tell the difference of the 2 pokes coming at you from the 2 toothpick a lot closer than you could on your back. Okay, almost I think 5 or 7 centimeters is when you can no longer tell whether the person's poking you with just one toothpick versus 2 toothpicks, because they're activating the same sensory receptor. Okay, all right. So the simplest type of sensory receptors we have is called the free nerve endings. And we kind of talked about this, where the neurons, dendrites themselves, are receiving the stimulus directly, just like those free nerve endings within the epidermis, as you can see in this picture right here. So these are those free nerve endings within the skin. And just like how enzymes are specific for their particular substrate, some sensory receptors are also only specific to specific stimuli, okay, as determined by the structure of the receptor or cells, okay? Like one type of enzyme can only bind this substrate A, and then another enzyme have to be used for binding the substrate B. Well, same thing for sensory receptor. Sensory receptor for pain only binds certain neurotransmitter, whereas sensory receptor for pH level binds to other stuff within your body, probably hydrogen ions. Okay, so that's how your body can tell the difference between different stimulus by having different sensory receptors that's specific for their whatever particular chemical they have to be able to bind to. Just like how we talked about with olfaction, they're sensing odorants. Well, odorants on taste buds probably won't do anything, okay, because there are different type of receptors, okay? And kind of like if you think about it as an example, just like how our receptors within the skin is not for tasting or smelling, we can put as much of that, for example, yummy ice cream on your skin, pour as much of it on your skin as you want, but you can in no way start to taste or smell that ice cream on your skin, okay? Because we have different type of receptor on our skin, because our skin is only specific for certain stimuli of their own, like temperature and pressure. You probably can feel the coldness of the ice cream and the pressure of that pile of ice cream on your arm, that gross dripping feeling dripping from your arm, but you will never be able to taste those food chemicals coming from the ice cream, because there's different type of receptors on your skin compared to your mouth. Okay. And as we have already mentioned, some of the pathway of signal propagation in chapter 17 for special senses, this is like a review on the process of transduction, going through what we went over already in chapter 17. You always start with a stimulus that arrives at the receptor cell, to induce a membrane potential change, either in form of a graded depolarization that may lead to an EPSP, excitation, or graded hyperpolarization that may lead to an IPSP. So what is happening within the receptor cell is referred to as the receptor potential. And once the stimulus is able to activate an action potential that leads to release of a neurotransmitter to the sensory neuron, this will then activate either depolarization or hyperpolarization within the sensory neuron itself, called the generator potential. And those arrival of action potential from the sensory neuron toward your central nervous system is what then allows our cerebral cortex to interpret those sensory information coming up and send out the appropriate command in response, okay? So now, sensory receptors can actually be divided into two main types depending on their ability to adapt. What do we mean by that? What do you mean by adaptation of your sensory receptors? Well, kind of like you walking into the anatomy and physiology lab on a dissection day, the moment you step into lab, that pungent odor of animal tissues, preservatives, comes in through your nostrils. And many of my students will make a face when they walk in the door and go, ooh, what's that smell? But after being in the lab for maybe 10 or 15 minutes, somehow the smell doesn't bother you as much anymore than when you first walked in. And some students can hardly smell it after a while. Well, that's because our sensory receptors for odorants have the capacity to adapt. Once the same odorant has been activating the same sensory receptors, it eventually results in reduction in the sensitivity of the stimulus. Just like how some individuals aren't aware that they have sprayed too much perfume on themselves because they think they don't smell the fragrance anymore and then ends up spraying even more on. But when someone walk by you, on the hallway, that person will be like, wow, that's some strong perfume, because that person has not been smelling the fragrance of the perfume all day, only you did. So that's why we are, sense of smell kind of adapt to the smell that we are, the odorants that we're smelling. And hence, tonic receptors are those that are always active, which means they do not adapt. It is, they adapt very slowly. It is the increase or decrease in the frequency of action potential that will reflect the increase or decrease in stimulation. So that's why you have this graph here representing tonic receptors, where you have this constant signal, normally action potential, action potential, action potential. And then all of a sudden you see this, intense in intensity of the action potential. Also thousands, tens of action potential, and then you go back to normal action potential, action potential, action potential. So this is saying that particular receptor is on all the time, receiving those constant action potential, and what it is detecting is whether you have increased amount of action potentials arriving at that specific receptor. Now, on the other hand, phasic receptors are those that are not usually active, and stimulation results in a burst of action potential that will end when the stimulus stops or if the stimulus doesn't change anymore. So, in a sense, according to this graph here, you see that there are absence of action potentials under normal situation, and also during this time, while the stimulus isn't changed, eventually it will then go back to no action potential when it adapts to that particular stimulus. So you can easily remember tonic versus phasic receptor, because phasic receptors will phase out. It actually phases out in its effect. Which means it will adapt, and no longer can you smell that particular smell coming through. And so the reason why phasic receptors have action potentials that phases out is because of their capacity to adapt very quickly, like what I was talking about your sense of smell earlier. Or kind of like if you wear socks, in the morning, the moment you put your socks on, you can feel your socks on your feet. But after, some time, you can't even tell that you're wearing socks. You're feeling those textures of the fabric while you're walking throughout the day, is because you are used to that particular feeling, and it no longer is sending that electrical action potential toward your brain that you have socks on. So fast adapting receptors are what phasic receptors do. phasic receptors, and the receptors respond strongly at first, but the activity will gradually decrease. For example, like change in temperature. So, for example, when we walk into Costco from our normal Texas summer heat of over 100 degrees, some of you, the moment you walk in, you're like, oh, this is nice. Or some of you may go, ooh, this is chilly, and you rub your arms and feel the goosebumps that's initiated by your erector pili muscles within the skin. But after walking around a few minutes in Costco, then those feelings are gone because your body eventually got used to or have adapted to that temperature. Okay, so that's what fast adapting receptors mean, and that is what phasic receptors do specifically. On the other hand, tonic receptors does what's called slow adapting, okay, that's why we call them slow adapting receptors, where there is almost no peripheral adaptations at all, okay, so no or little adaptation. For example, when we get a cut on our fingers, it feels painful when it was cut, okay, pain, it turns out, is a type of tonic receptor. So when you first cut it, it feels painful. But after 5 minutes, it still feels painful, okay? And sometimes even more painful 10 minutes later, okay? Because now that you can, maybe that area of injury starts swelling, starts undergo inflammation, and you feel even more painful. So you end up, you will continue to feel the pain until either you rub some medication on that will help you get rid of the pain because of the chemical may block some sensory receptors or something that will alleviate you of the pain, or when the wound starts to heal is when you no longer feel the pain. But you don't get used to pain sensations to the point where you no longer feel the pain. Like, I can keep cutting my fingers, but it will hurt every single time it gets cut. I won't get used to that pain. Or if you think about women giving birth, then if pain was a phasic receptor, then women probably don't need epidurals during labor and delivery because we can get used to the pain. That would be nice. I would have liked that, but that's not what happens. You don't get used to that pain, and that pain unfortunately gets more intense as the uterine contraction gets more frequent. And, if you same thing with not just pain, but, same thing with photoreceptors. Remember those photoreceptors on your retina we talked about in chapter 17 for their reception of light. So if those receptors in our eyes get used to the constant light signal coming through to the retina, which is constantly because we're constantly looking at stuff, which means light is always being bounced off onto the back of our retina, then if photoreceptors are a type of phasic receptor, then we're in trouble. We're going to get used to that light, and then that signal is going to be gone, and ends up that we're probably going to be momentarily blind throughout the entire day if photoreceptor was a type of phasic receptor. We cannot afford for these different receptors to be a phasic receptors, or else, for example, for pain, we wouldn't know when we're putting our bodies in danger, or for eyes, photoreceptors, we would go blind. Okay, so there are reasons why some receptors are tonic and why some receptors are phasic. And hence, that's what slow adapting receptors are. They are those that cannot adapt and will be consistently on, just like your photoreceptors and pain receptors. And, what changes is just the intensity if a suddenly stronger stimulus arrives. And that's why we can see brighter light versus dimmer light, or really, really painful situations versus less painful situation. We are able to feel all degrees of those all the time. Okay? All right, so there are 4 main classes of sensory receptors. They are your nociceptors, thermoreceptors, mechanoreceptors, and chemoreceptors. We may have slightly mentioned a few of these, but we just didn't really give those receptors an official classification name. So it shouldn't be too hard with these, okay? So nociceptors are those that are for sensing specifically pain. okay, which are common on superficial portions of your skin, joint capsules, periosteum of your bones, around walls of blood vessels. And those are, places that are more likely to have injuries. And if we don't feel pain, we can continuously injure those areas, which we don't want to do. So that's why we have nociceptors in those particular areas of the body. And from what we talked about, earlier, nociceptors are tonic receptors. Okay, so tonic receptors. Now, they are usually free nerve endings when we talk about nociceptors, and they usually have a large receptive field, which explains why sometimes it can be difficult to determine the exact location of the source of pain, especially when we think about internal pain. For example, when you go to a regular physical examination and you feel somewhere that's painful and the doctor asks you, where are you feeling pain? You will likely answer, well, somewhere in my chest area and somewhere in my tummy area. But you won't be able to tell your physician accurately that, oh, it is my right ventricle of the heart that leads me to feel this pain, or it's my transverse colon that I feel something is stuck in there, so I feel pain. You don't say it like that. It's because these nociceptors all over our body usually have large receptive field that we just kind of know the gist of where that pain is coming from, especially, like I said, when the source of pain is coming from an internal part of the body. Your skin may have smaller receptive field than internal. And if that was the case, technically speaking, if we have very small sensory receptors where we can tell our physician exactly where or what part of our body is in pain or what part of the organ is in pain, then no one would ever need to do X-ray or MRI scans. The whole point of doing those scans and those imaging is because we want to know exactly which bone that's fractured so we can help, fix it up, okay? Or what part of the brain has lesions, or what part of the intestines have something stuck in it, because you can't tell me that because you can't tell your physician that because we don't have those tiny little receptor fields or receptor cells that tells us exactly where it is in our body that is the source of the pain stimulus. All right. There's also a difference between fast pain that we feel versus slow pain. Fast pain is, for example, when we get a vaccine injection or have our blood drawn for a blood test at the clinic. This is that very quick pain. You feel it very quickly and then it's gone when the needle goes into you. As soon as the needle pokes in you, you feel that little pinch and then, they pull it out and then that pain is gone. Usually we can locate the source of pain at least several inches of the receptive field. If the nurse is poking at you on your forearm versus poking at you at your thighs or something, okay? You know the difference. Now, slow pain, on the other hand, tends to be more associated with burning or aching pain, which the receptive field can be even larger for this type of pain. Like I was saying, we usually feel pain in internal areas, but only have a general idea of where that pain is coming from, like your chest, abdomen, thighs, etc. But you can't tell what part of the chest specifically, maybe middle of your chest, but you can't tell which organ is it coming from or which type of tissue is it coming from, okay? All right, and the neurotransmitter that is released by these sensory neurons, that relay the sensation of pain from your nociceptors to your central nervous system are glutamate and substance P. Substance P for pain. How nice. Okay, so substance P has a P in it, so it is for pain. Glutamate, you just have to remember, okay? And there are neuromodulators called endorphins or enkephalins, which are able to inhibit their release of these glutamate or substance P along the pain pathways. Remember, we mentioned neuromodulators compared with neurotransmitter from chapter 12, okay? Neuromodulators are those that control the amount of release of neurotransmitter from the neurons. And these endorphins and enkephalins are actually structurally similar to morphine. All right, the second class of sensory receptors we have are called thermoreceptors, which are free nerve endings located in the dermis, skeletal tissues, skeletal muscles, liver, and your hypothalamus as well. And they're usually very active when temperature is changing, but they will quickly adapt to stable temperature, which means, are they tonic or phasic receptors? They are phasic receptor because the feeling phases out. Okay, so they adapt, they phase out once you're used to that particular temperature. Okay, now I'm not talking about like putting your hand in boiling water and you're going to get used to that boiling water temperature. Okay, I'm talking about normal, within a few degrees range, like 37, or like if we were talking about Fahrenheit, from 70-degree Fahrenheit to 60-degree Fahrenheit. Okay, I'm not talking about boiling temperature. Your hand will burn off. Okay, all right. And then mechanoreceptors are the third main class of sensory receptors. They are those that are sensitive to physical stimuli that usually involve distorting your plasma membrane, which, it could be pressure, sense of position, or sense of touch. And we already mentioned some of these from the integumentary system. So there are 3 different types of mechanoreceptors. And like I said, we already mentioned Meissner's corpuscles, Pacinian's corpuscle, free nerve ending within the epidermis. Those are considered as tactile receptors, which are sensitive to fine touch or pressure or even vibrations. Okay, so they usually provide that sensation of you touching something, feeling different texture, or if someone is pinching your hand or compressing, sitting on your hand, you feel the pressure coming through, or if you're sitting on a massage chair, you feel the vibration that's coming through. Okay, so there are some that are specialized for that fine touch and some that are specialized for crude touches that we talked about in the integumentary system. Your book actually goes through 6 different specific types of tactile receptors, but we won't discuss all the different specific types here. We have already mentioned a few of these already from the integumentary system. For example, like the root hair plexus, that's a type of tactile receptor as well. All right, so the sensitivity to tactile sensations may be altered by infection or diseases, though, or if there is a damage to the sensory neuron or pathway. So that's why when we lose our sense of touch or vibration or pressure at certain parts of the body, this gives us a good clue to allow us to map that tactile responses that can aid in clinical assessment of what's going on. Okay, if you lose feeling in a particular part of the body, maybe it has to do with certain other spinal, other part of the body, like what we talked about with dermatome in chapter 13. Okay, dermatome is that bilateral surface area that a particular pair of spinal nerves serves. So the fact that you no longer feel someone touching you on that particular part of the body, a particular dermatome, for example, then we can have some clues as to what's going on within your central nervous system. Okay. So a second type of mechanoreceptors are called baroreceptor. If you know what barometers are, then you know this type of mechanoreceptors monitor in the body. Okay, so barometers are those that measure pressures. So baroreceptors are the ones that are monitoring pressures within your body. They consist of free nerve endings that will branch within your elastic tissues in the wall of a distensible organ, whether it be stomach, intestine, blood vessels. They all have baroreceptors to sense pressures. And that's how we are able to monitor our blood pressure levels to maintain homeostasis, make sure that our blood pressure is somewhat close to 120/80, and hence whenever someone has low blood pressure or high blood pressure, that probably means something's going wrong with your homeostatic mechanism, that you cannot bring that down back to normal temperature, up or down back to normal blood pressure. Okay. And that means an existence of a disease, which was how we define the inability to maintain homeostasis. That's what results in being sick or a condition that's a disease, okay? So how we can monitor our lung expansion is how we can set the pace of our respiration, okay? For example, if we have our baroreceptors within the lung says, we don't have enough oxygen, okay? So that's going to actually get your respiration to be faster so you can breathe harder and quicker so you can get those oxygen in. And we'll talk about this stuff, what happens when you're low in oxygen or high in carbon dioxide within the respiratory system next semester, okay? And for digestive and urinary tracts, there are stretch receptors that will sense the pressure of food particles that are entering through your GI tract or your bladder, okay? So if a lot of feces is accumulating within your last part of the colon, that will trigger a reflex of defecation. Or if there's a lot of urine within your bladder, that's going to trigger urination. And how your body can sense that there's a lot of stuff in that area is by these baroreceptors within these hollow organs. Okay. And the third type of mechanoreceptors we have are called proprioceptors, which are usually found in joints, tendons, and muscles. Now, not in the organs of thoracic or abdominal cavities. That's why we can't sense where our heart is or our lungs is. We know where it is because we know human anatomy, but we can't feel where our heart is unless we're feeling for the palpitations of the heart. We can't feel where the lungs are or where our stomach is, okay? So we're talking about when we have proprioceptors, they're, what's monitoring your position of joints, tension in your tendons and ligament, and your state of muscular contraction. So an important role for proprioception is, for example, stability. Being able to stand up straight or the ability to walk require our self-awareness of where our limbs are. We must continuously monitor our posture and adjust our muscle activity as needed to improve balance. So this is why if you're concentrated on other things like reading a long text message on your phone or reading a book while you're walking or running, you may be more likely to accidentally trip and fall because your brain is focused on other stuff versus your proprioception of your limbs as you're walking, okay? And even something as simple as brushing your hair or throwing a ball into a basket, to what degree do you move your limbs to be able to aim at a certain target is made possible by proprioceptions. The fact that we can practice throwing basketball hoops, okay? And eventually you get the hang of wherever you're standing, that's how far your hands got to go or how hard your hands and muscles got to push in order for the basketball to make it to the basket. So it is all part of our awareness of where those body parts are or tensions within the muscles. Okay, so the last class of sensory receptors are chemoreceptors, which are the ones responsible for sending chemical status information toward the autonomic nervous system concerning respiratory and cardiovascular functions mainly. Now, there are sensory information that we may not be consciously aware of, okay? And this is one of those. We cannot sense how acidic we are, okay? We cannot sense how alkaline we are, okay? Those are monitored by chemoreceptors, but we cannot consciously be aware of how acidic our body is or the fact that we're lacking in oxygen. We may feel like suffocation if we don't have enough oxygen, and a little bit dizzy, but that is not a measurement of how much oxygen that's in your body that you can feel, like, pain. The more pain you feel, then there is more of that, injury that's occurring. So this is a type of sensory receptor we are not consciously aware of, okay? So for example, chemoreceptors in our respiratory centers of the upper medulla oblongata is sensitive to changes in the pH or carbon dioxide content within the cerebrospinal fluid, which then, if there are perturbation, okay, that will perturb the pH level or CO2 level, this will trigger a reflexive adjustment in how deep you're breathing and how fast you are breathing. And like I said, which we'll talk about next semester, okay? Chemoreceptors within carotid bodies or aortic bodies, of course, has to do with your blood vessels, okay? So they will be sensitive to changes in pH, carbon dioxide, and oxygen content within the blood that will trigger reflexive adjustment for your respiratory or cardiovascular activity when these things differ. Which then, that's why next semester we'll talk about, respiratory acidosis versus metabolic acidosis, or respiratory alkalosis versus metabolic alkalosis. All those things are due to the presence of chemoreceptors in different parts of our body. All right, so the somatic sensory pathway can be divided into 3 levels of neurons. So the first order neuron is the sensory neuron that delivers the sensory information directly to the central nervous system, either through a cranial or spinal nerve ganglion. The second order neuron, which can be part of a pathway too, because not all pathways are monosynaptic, like we talked about. A lot of things we can, sometimes we need an interneuron in between to relay the information. So interneuron is known as a second order neuron that can be residing within the spinal cord or the brainstem. And then the third order neuron is what the second order neuron must pass through in order for us to be aware of the sensation, which usually involves the thalamus other than your olfactory sensation, like we talked about in last chapter. In chapter 17, olfactory information goes straight from your first cranial nerve into your cerebrum, okay? But for other than that one, all senses has to pass through the thalamus in order for you to be consciously aware of that sensory information coming through. So this diagram here draws a good example of what happens when we have, if we're able to be consciously aware of a sensory information coming through. For example, if we're talking about hearing, or our ability to receive auditory informations, hair cells within the cochlea, which are the receptors for sound, must be activated to send electrical impulses to our first order neuron, which is probably your auditory nerve, particularly your cochlear nerve, okay? That is your primary afferent fiber, which then will go on and move the information toward your second order neuron, which will be within your brainstem or spinal cord nuclei. And for cochlea, there are a particular area called the cochlear nucleus within those areas. And then they have to pass your third order neuron, which should be residing within the thalamus. And for hearing, they have to pass through something called the medial geniculate body, which then at the end, in order for us to be able to to sense and perceive that sound coming through, it has to get to our auditory cortex, which is a primary sensory cortex for our sound within your temporal lobe of the cerebrum. Okay? All right, and decussation, as I have mentioned before, is the ability for axons of usually the second order neuron, which is our interneurons, to cross over to the opposite side of the central nervous system. So for example, the right side of your thalamus may receive information from the left side of the body, such as someone touching your left arm, but the information can go into your spinal cord, up to your brain on the right side by decussing over to the other side. It's kind of like this example here, that information is coming in from the person's right side, and then it goes up to the left side because this particular interneuron relayed the signal and has done a decussation over via the gray commissure of your spinal cord, which then goes up towards the left side of your thalamus. Okay, so your textbook actually goes into detail about what type of sensation goes through which specific tract of the white matter within the spinal cord. Remember, we talked about that, how white matter of the spinal cord have ascending tracts versus descending tracts, and that's pretty much where we stopped at. But the book actually goes into more details. There's different labeling of different tracts around the white matter of the spinal cord that are for different senses specifically. But again, since we don't come back to these concepts in the next semester, I decided to not go into details about those particular parts. Okay, just know what levels of neurons you can have and what does decussation mean, okay, for this part of the somatic sensory pathway. So any abnormality along the somatic sensory pathway can result in inappropriate sensations or inaccurate localization of the source. So those receptor pathway we're talking about, if something was interrupted or something went wrong, we can have two different types of condition. For example, there's one condition called the phantom limb syndrome. And as the name suggests, sounds kind of scary, phantom. Well, that has to do with a limb that has been amputated, and somehow the person can still sense that there's that limb there still, even though it's gone. So it's usually caused by activity in the sensory neuron or interneuron that were once part of a developmentally programmed pathway in an intact limb. So for example, our sense of proprioceptions we talked about. We know that our right arm is right here and our right foot is right there. I know it's there because, I was born with that. Okay, I know I have those features on my body. Okay, but all of a sudden you amputate off the arm or amputate off the foot. Somehow the brain still senses the existence of that proprioception of that particular limb. But visually speaking, you don't see the limb there, but you're like, no, I feel that particular foot itching and I want to scratch it, but it's not there. Well, that's because there are, that's why the hypothesis out there is that there may be some type of memory that is associated with proprioception, that our certain limb or part of our body is there. And it takes a while for the brain to get used to the fact that that limb or that part of your body is no longer there. And you just have to, get used to that idea, because the proprioception kind of have a conflict with your visual sensory information, because you no longer see the limb there, but you feel it there as if, okay? All right. And referred pain is another concept that stems from how these somatic sensory pathway works. Sometimes pain is felt in an uninjured part of the body when pain actually originates in another location. So, for example, for heart attack patients, when they feel a heart attack coming, rarely do they feel this part of the body or this part of the body hurting. They feel that it hurts around your shoulder area and it radiates down your left arm. Okay, so just because it is your heart that's experiencing something going on, your heart may not hurt, but what starts hurting is your shoulders that goes down to your arm, but is not something within your arm that's going wrong, but is your heart. So that's what referred pain means, and hence, which is why I talked about with that, physical examination. If you're trying to tell your doctor where it is painful, it may be a referred pain that is not that particular of the body that is painful. That is something going on. Okay, so. For example, here, if it's your liver or gallbladder that is hurt, that something's going wrong, sometimes your right shoulder can be painful. Or whenever we feel stomach pain, we know we don't necessarily feel pain on the left side of the body, but sometimes we feel it right in the center, or this whole tummy part will feel very painful when something's going wrong with our large intestine. But our large intestine is actually not in the center of your abdomen. It actually goes around around that area. Okay, you have ascending colon, transverse colon, descending colon, and a sigmoid colon before you get to your rectum. It actually goes around that area. But yet when something's going wrong with your colon, you feel pain in this general abdominal area instead. So that's why it's a referred pain and not directly where your large intestine is where you will feel pain. Okay. All right, and these type of referred pain can be explained by, pain sensations that can be arriving at a segment of the spinal cord that also gets stimulated with the interneurons that's within the same pathway. And that's why we experience pain, because as the signal is coming through toward your central nervous system, it might activate those other neurons along the way too, interneurons that can connect to those other places of your body that makes you feel pain in that particular area. Okay. All right. So that is all for the somatic nervous system that we will discuss, and we will move on to the autonomic nervous system, which is all the responses that happens within our internal organs that our central nervous system controls for us without being aware of all that is happening. And I think this should be a good time for us to take a break, and we'll come back to discuss the autonomic nervous system and memory in part 2 of the video.